más o menos. Hello, good afternoon. Thanks for coming today in this webinar from the SEBBM. We have Javier De Las Rivas. Javier is the coordinator of the group that now it's called Biología Molecular, Ómica y Bioinformática. And he is from the Cancer Center of Salamanca. He is a professor of research. And so, Javier, thanks for coming and giving this talk today. And well, when you want, you can share the presentation. Remember, please, that you can send questions through the chat, and then I will read them and I will ask Javier what you have in mind. Thank you.
Okay. Can you see my screen?
Yes, now, yes, perfect.
Okay, thank you. Well, thank you to the SEB for the invitation to give you this talk of the work that we are doing now in my lab in Salamanca in the last year or so, especially, that is focusing the analysis and development of tools for the analysis of single cell data. And this work is not done by me, but by my group, especially here by two persons that I will explain later, that are Natalia Alonso and Enrique de la Rosa, that they are doing the PhD with me in Salamanca. Let's see if I can. Okay. So, in our study of the different cell types, we started in a strong collaboration with in cancer with oncohematologists and with hematologists, and also in the study of the microenvironment of the tumor microenvironment, where all the hematological and immune cells are critical. So the first approach that we took when we started to apply the studies on single cell data is how can we see the different cell types that are in this complex tree, of the hematological hematopoiesis tree, this complex system that is our blood and an immune system. How can we distinguish the different cell types and which one are present in the tumors or which ones are present only in the microenvironment around the tumor and things like that, no? So this was our first approach to study a lot of data related with mainly with bone marrow samples. Here, can you see my pointer? Okay. Yes, the bone marrow samples, no? As you can see in the cells, there are these two main lineage, the myeloid lineage and the lymphoid lineage, and they have very different cell types. And our interest was, can I pass? Yes. Our interest was to see if we can find specific markers when we analyze single cell data generated from peripheral blood or bone marrow mononuclear cells that are mainly the cells that are in this hematopoietic system that are not the erythrocytes, all the ones that have nucleus, one nucleus. It is a typical part of the separation of cells in hematology, in bone marrow. That is to produce the mononuclear cell fraction. and see if we can recapitulate the markers that has been assigned to these different cell types in the single cell data. This is not an easy task because, as you know, the number of cells here are many different cell types, and also the concentration of the cell types is quite different. For example, you can see here if we compare one part of the whole blood, the peripheral blood or whole blood. When we go to quantification of the different cell types, lymphocytes is 27%, or monocytes is only 8%, or neutrophils and eosinophils is nearly 60%. But it is known that, for example, the technologies, the single cell separation, didn't manage to obtain good numbers on neutrophils when they do the single cell separation, because the neutrophils are due to the shape and things like that, no, and how big they are, no. So our approach was try to see if we can separate the standard, different standard cells of the different T cells, B cells, monocytes, dendritic cells, and so on, and also identify the typical, if we can identify in the transcriptomic profiling observed by the single cell data, these specific CD markers, no, that are well known by flow cytometry or by protein antibody markers, no, like the CD4 for T cells, or CD8 plus for another subtype of T cells, or for B cells CD19, and so on, no. And this was our first attempt, no, to identify the different, and especially because we participate in a European project focus on myeloid, the myeloid lineage. Why we had a special interest in the myeloid lineage? Because the lymphoid lineage is much more studied. The B cell, T cell, NK cells is easier to isolate, and there were a lot of data on single cell, but not so much because of the lack of observations of different cells of the myeloid lineage, especially, for example, the dendritic cells or macrophages that are very big cells, or especially macrophages or monocytes or granulocytes in general. They are not very well populated when they do this technology, when they apply the technology of single cell separation. And as you can see, for example, for dendritic cells, there are several papers. One of them is this, but that have characterized not only just the general canonical cells, B cells, T cells, dendritic cells, but, for example, now with the CyberSOR system that has been very much dedicated to these hematological and immune cells, there are at least 9 types of T cells, no? Or you can see here in this review, well, in this work, the different types of dendritic cells that they identify with these different characteristics and different concepts coming also from the both lineage. That is something special of the dendritic cell, can come from the myeloid lineage or from the lymphoid lineage, and this makes the system complex. So to address this idea of identifying the cells that are in our single cell data, we work in 2 approaches. One was applying the convolution methods on a lot of RNA-seq data that they have very good transcriptomic profiling of bulk data of these samples, and also going into development of methods of single cell RNA-seq more specific, no? The first part of my talk is about this, how we apply deconvolution methods to analyze what is called what is bulk expression. Well, bulk expression is what all we know is that the RNA-seq, no, the RNA-seq itself. We have for example, of a tissue, of blood or bone marrow, all the tissue cells go there together, no, and so we have a transcriptomic profiling of all the cells at once. The idea, can we design methodologies, bioinformatic methodologies that produce what is called a mixture deconvolution, cell mixture deconvolution in this case? And this is what we address with the work of Natalia, that I'm going to put this. And with the work on Natalia and in the work, let me move this a little bit. Okay. That's where what we do is we take bulk RNA-seq data from different data sets. I am going to tell which ones, and then we apply different deconvolution methods to try to identify if we can identify the different cell types that are there in this mixture and what is the proportion of each cell type. So to do that, we need two things. We need a gene signature matrix that tells me that these genes provide a good signature for a given cell type. And I also need, in a second part for this analysis, I need the bulk expression signal. That is the expression of RNA-seq, no? The RNA-seq global of the mixture. Doing this, what we do, well, of course, the gene signature matrix, we can get it from experience, from knowledge, or from different methods. For example, we can also obtain, nowadays, most of the methods go to single cell data on controlled data set where they know what the number of cells that they have, for example, because they separate in a control experiment, in a supervised experiment with flow cytometry, different amounts of different cells, and then they put them in the single cell system, and then they see which are the genes that better characterize those cells, no? For example, the B cells, as you can see, which are these genes that are specific and of the different cell types. This is what we need here, a gene signature matrix. And also we know later on we need a mathematical method that is usually is a method that is the composition methods of multiple matrix, in which we want to identify what are the cells present in the mixture of the bulk signature, bulk signal that we have, no? In this way, what we, of course, to start this approach and getting different deconvolution methods, what they are called mixture deconvolution methods, we didn't just started from scratch. What we did for sure is go to some methods that are well known and compare them to see how they perform, no? And in particular, we compare in this study 4 methods. That is CiBSource. CiBSource is very well known because having published with very important papers with the low quotations for, and it has got a very good signature for all the cell types of hematology and immunocells. Later on, and further, these two are supervised methods. What means supervised methods? Methods that need a gene signature matrix, and they need the matrix with the gene signature. Okay, but and the signature, that is the S here, this one. And then the conical linset are methods that they are unsupervised. What is that? Is that they don't need the signature matrix. They calculate what are the genes that better are assigned to the different cell types. What is more difficult to do and computational, many times not very efficient, but we try to compare those methods. So, and what we did then is to start with 4 samples, data sets from RNA-seq. All this is bulk transcriptomic profiling from microarrays of Affymetrix or RNA-seq or microarrays from Illumina from different samples, where we are always, as you can see, identifying, the samples are peripheral blood mononuclear cells, and in one case, that is a control experiment, we have also they preparing in a mixture, control mixture, they added cancer cells. Those are colon cancer cells, just to put in mix with the other cells and to see if the system recognize the cancer cells from the other cells. So in the analysis of this, what is the first challenge? The first challenge is how many cells, those are the different data sets, as you can see, and this is our reference CiBSal with the LN22, sorry, that how many different cell types and subtypes do you want to distinguish? Because you want say, no, I want to go to I'm very much interested only in T cells. Okay, T cells, and how many types of T cells? 9, for example, these naive, CD4 positive, CD8 positive, memory, resting memory, all these regulatory follicular helper, you see 1, 2, 3, 4, 5, 6, 7, 8, 8 different in the our matrix of CiBSal, and CiBSal have a signature for each one of these T cells. Or we want just to identify lymphocytes, or we want to identify T cells and B cells. That's a very important first step, because it's the level of granularity that you want to accomplish. And this is critical because with in the different, for example, in these different data sets, you can see the number of cell types that they identify is different. In this, for example, is only 4, here 5, 11, 17. So this is a first decision that you have to make, and also the method can be very good when you have the granularity is not too deep. For example, you go to 10 or 9 different cell types or subtypes, but when you go to many more, maybe start to fail. For example, as you can see here, in most of these data sets, we didn't have macrophages or mastocyte cells. And for example, as you can see here, in neutrophils, some of the granulocytes in some of the data cells are not very well populated, what I said before, and they are very important usually in these samples. So what we did is to see, okay, because we have for these data sets, we have the known data, because we have a control experiment, and we are applying 4 different methods. Now what we can do is to compare the methods, how they perform, how is the predicted estimated proportion of each one of these cells, and how is the real known expected, because has been experimentally provided by a supervised method. For this reason, we took one of these examples that I told you before. And then, as you can see here, Fibersor, all of them in general for the main these main cells, they perform quite well, no? B cells, T cells, neutrophils, monocytes, NK, for example. But we observe that some of them have uh, um, here is the estimated value of number of cells corresponding to this cell type, and this is the real number. And then when it is one, this is one, is that the real number is equal to the estimated. No? So in this way, we perform this kind of correlation plots to see how is the performing of the methods. In general, when we do this comparison globally, the methods perform quite well. But when we go to more deeper analysis, for example, here we got Fibersor and FARDeep, the two supervised methods that they have a data matrix. You can see that FARDeep, sorry, um, uh, in both cases, the well here we are estimating the different these 5 different cell types in each one of the samples. Each one of the samples, and here is the predicted in dark colors, and in most pale colors is the known value. And as you can see, for example, Fibersor, here we got a sample where these two samples, what happened to these two samples? Why the real says nothing about these data sets? Why? Because in this data set, we put these cells that are cancer cells from a colon cancer cell line that are not immunological cells, but Fibersor is estimating that they have some immunological cells, and these samples, the sample 1 and sample 2, are pure concentration of what uh, mm, cancer cells, as you can see here. You see, this is the cancer cell that was added, and now we have 6 different samples. And then now in the dark colors are the estimation, and in the pale colors are the real data. As you can see, Cibersol always estimate that the system has got, despite the fact that these two samples are pure cell line, estimates that it has got that. Why? Because Cibersol is not designed to identify cancer cells. It's designed to identify. So when finds cancer cells, it gets muddled, and the result is not good. For example, Linset in that sense was much better, saying no, in these two data sets, in these two samples, you have a very good estimation of that is only one cell type. And in general, the proportions calculated by Linset are, you can see the error here when the root mean square error we calculate in this way the distance when it is 0 is perfect. So here the numbers are for Linset smaller than for Fibersor for the same samples. Another way of measuring this is this kind of percentage, no? Percentage of a given cell, for example, NK cells, B cells, B cell, NK cells in different data sets, in different samples, sorry. And in the real data is the red, one, and the this blue is the estimated. And you can see that the Konica, for example, does a very even estimation of demand was always the same, and we discovered that the conical is not a very good method when you illustrate with this way. Linset, for example, that was the one that performed very well here, better than Cibersol, also performs quite well here, as you can see, estimating, because the red is the estimation, how many NK cells do we have in the two first samples, none, and estimates none. And then here you see the trajectory that is plot for a given cell, NK cells or B cells, is very good in general. Again, the correlation high and the root mean square error in proportion also quite low. So in this way, we compare different deconvolution methods. We try to understand how we interpret the results of these methods, and also how can we compare them. And in fact, in this work, what we did is to design these kind of ways of comparison, these three ways of comparison, with this kind of plots that I showed you, that are quite useful. And this last is a comparison of the Linset method that was on our hands the one that performs better. And even we took the Cibersol matrix that is very well known and used in many studies on single cell analysis of hematological immunocells. The LM22 is called like that because it has got 22 different cell types identified, mainly, as I said, immune and hemato. And then what we managed to do with Linset is to provide for these five major cells a signature of cells of genes, of 170 genes that are better characterized, that are the ones that are selected by Linset, that are better characterized for these cell types. In fact, you can see here the number of genes that has got Cibersol in total has got 547 genes in total, and that is in this, they are here in this heat map. But with Linset, we managed to get a better, or even better, for these five major cell types separation with only 17 genes. So, sorry, 117 genes. In this way, we managed to get a more specific signature. That this was one of our scopes in this work. Okay, we published this in a paper, and Natalia is the first author. And if you have any question, you can ask me about that. And sorry that I have a little bit of a cold. And the second part is more focused on the analysis of data of single cell. So with the work of Enrique, what we started to do is to develop a complete pipeline of analysis of data from samples of single cell data, mainly focused on 10x genomics, because 10x genomics is the technology that has been most used to obtain the single cell plots. And then to do that, we work again with, because we know well there's a system quite powerful to identify different cell types, with a peripheral bone marrow, or peripheral blood, sorry, PBMC, that are peripheral blood mononuclear cells, or bone marrow mononuclear cells. This and this, okay? 3 data sets. And in the first data set, again, we have a very good control because these people, when they publish this paper, they already did all the flow cytometry data to calculate the real concentration of the cells. So we know what is the real result in this data set. So it helps us a lot to prepare this protocol of analysis. As I told you before, what is essential in any single cell data analysis first is to see how many cell types I am going to analyze, what is going to be my granularity, my level of separation of different cell types. And as you can see here in this plot that we prepare here, taking again this kind of cells, myeloid lineage, progenitors, and lymphoid lineage, that is more complex than before. We have the possibility of identifying 7 different cell types, 16 different cell types, and 27 different cell types that we try to plot in this complex system, which are the cell types that are at the different levels of granularity that we could manage to study. And also, because we wanted to identify gene markers, we took also 2 things. The CD markers, CDs that are well known as cluster of the differentiation proteins that are well known in UniProt, that is a collection of 369 genes. We took all the from UniProt human proteome, all the genes that correspond to a membrane protein, probably in the cellular membrane. For that reason, good candidates to be also to become a 3D marker of the cell surface. And also, we in a blind system, we took the top markers of the data sets without any kind of preselection. So we started the analysis with these 4 data sets. As you can see in the different data sets, we have, for example, in this one, 27 cell types identified, in this one 19, and in this one, I think it's the one that is 7. Okay? So why is that? Because it's the way you prepare the analysis, no? And then, and also when you put the data on this human plot, the transcriptomic profiling of the cells provides a given number of clusters, that's the clusters, automatic clusters, that are in theory corresponding to different cell populations. For that reason, we say 27, we could say 27 cell population, or 19 cell populations, or 7 cell population, you understand? And those are the plots originated just with the CD markers, plus 63 membrane proteins that we identified that were very good markers, or with all the expressed genes. As I told you, the single cell data, when you have the transcriptomic profiling, the expression single cell expression data, to go into the UMAP or in this plot that represent the cell populations, also runs other algorithms that are important to see how many groups how many populations can I distinguish in these data sets, no? And this is an algorithm that is important to identify the number of populations that are different or separated or independent in the different data sets. As you can see, the granularity in this data set is very big, and in these other two data sets are smaller, but they are similar cells, so we keep a comparison. So once we have done the analysis of all these identification of the populations, later on the question is that, can you now identify for me the top markers in each one of these populations? And this is what is start to be important. For example, in a given, for example, here, the lineage, lymphoid lineage, no? We identify first, are we observing what it should be observed, not that the standard cells, T cells, are positive for some markers that are well known, especially in expression. For example, CD8A or CD3G. Yes. So this is good because those are well known, present in several cells and are observed well known, no? But later on, what we can do is with these subsets of the TSN plot or the UMA plot is similar here in our hands. The TSN perform better, so is to identify, okay, and which are some of the best markers for this cell population. You can see, for example, that have not been that are not, for example, CD markers. As you can see, CD markers already reported in the literature. For example, with TRAT1, we identified TRAT1, that is quite a good marker, as you can see here, of a population that has been here identified as a CD4 plus T cells. Or, for example, the other case here, in the other population, CLL TCL5 is typical of CD8 plus T cells. And the same we can do for other proteins, especially other markers, especially when they are not, they are coming from genes that are or coming from our subset of membrane proteins that best mark our data set, or from all the other genes, no? For example, UVCAM here is marking the classical monocytes there, or for example, SERPINA1 is marking non-classical monocytes. You see, and this is interesting because this is observed in one cases, and you can compare that the things are quite reproducible. In this way, what we managed to do is for all the sample sets that we were studying, that are all the different cell types, that we were studying, that in this case were quite a lot, we identified specific cell markers that in some cases are were well known, and in other cases were not so well known. To validate the power of these cell markers, what we did is a little bit of machine learning approach that is the following. We can take use the gene signatures that we are creating, no? The different signatures that we are creating, and then perform a random forest machine learning method to see how they predict these genes predict the correct cell types in each one of these data sets, in different with different signatures, no? The genes here in this where we have the top 30 unique CDs from the dataset 1, that is dataset 1 that is the one that was supervised, or the top unique CDs plus membrane proteins that we added these membrane proteins, or all genes unique, or fiber source. Here just we put fiber source comparison. As you can see here in the accuracy of the runs, when we did this run of this prediction, of course, you see what is this 7, 16, 27. It was, I told you, is the level of granularity, no? The level of granularity when you go deeper, the accuracy is reduced. But for example, let's have a look at to the middle here. The middle here with our dataset where we put all our genes, we have one of the best performance for 16 cell types with an 80%, 84% of accuracy. That is better than the accuracy provided by the signatures using CyberSoft, or by the signatures using only the CDs, standard CDs that are in the literature and well known, or the standard CDs plus the membrane proteins. So this indicates that we are discovering some markers that are quite powerful in the identification of some cell types. And even we can say of these 17, here's the case for 27 cell types. We go here for the comparison of the overall accuracy, the best accuracy provided by our method, and you see the identification all the different cell types that we identify. That is quite a lot different, no? Like many B types, naive B, a lot of different cell types that, in fact, all these are described at the beginning of our work, no? The different 27 cell types. In some cases, it's more difficult, but in some cases, our signature is quite powerful. And finally, what we can do also with our method is, okay, now we have a UMAP or a map of our population, cell population. Can we apply methods to identify trajectories based on the genes, where the genes move from one population to another in a coordinated way, to see if these trajectories reflect something in biology? And we did that. And for example, here, you can see this is a typical example of CD34 is a typical marker of hematopoietic stem cells that is very high, as you can see here in this green population. And then when we go try a trajectory towards, for example, to the blue data, this disappear. Why? Because this is a differentiation line. We will see. And what is appearing here in this line from hematopoietic stem cells to some specific lineage. What is appearing, as you can see, is CD20, or that is CD19 as well. That is a typical marker of B cells, as you can see there. So, in fact, these trajectories makes a lot of sense when you are able to apply an algorithm that runs well these trajectories. You can see there. And we did that for other trajectories, for example, knowing, and we identify very well that this now the here in yellow and orange color is the stem cells area, and from this area that are in our population later on. There are like three very interesting generations, no? The myeloid lineage that goes like that, the myeloid lineage, and here at the end, what we have here, the monocytes, the different types, classical monocyte, pre-monocytes, classical monocytes, non-classical monocytes, all those are here in this lineage myeloid line. Or, for example, here we have the lymphoid lineage of the B cells or the lymphoid lineage of the T cells and NK cells. And you can see that is clear as well, where we can identify markers that mark this evolution of trajectory and pseudotime. Some people call this pseudotime. I don't like these pseudotime things, but at the end is how we move from hematopoietic stem cells to B cells or plasma cells in this line that is well known, and all the cells are very well located in these trajectories according to the gene profile. And this has been published here in this paper at the beginning of this year. And if you have any question, you can ask me. And that's it.
Thank you, Javier. Thank you very much. I don't know if we have questions yet. No, it doesn't seem to me.
Can we stop sharing maybe?
Well, I think this is really interesting. Yeah, there's something that we will all need. This is like we all try to do single cell or so on, but even if we come from another field, like for example liver diseases, no, that's where I work, no.
Yeah.
I have it's outside this kind of cells, but with your system, do you think that we could know the trajectory of a parasite that will become a tumor cell? Because in hepatocellular carcinoma, it's a kind of cancer, of liver cancer, that comes from the hepatocytes. So I was wondering if with this kind of methodology, we could be able to know which hepatocytes are going to be malignant in that progression of the disease.
Well, if the question is that you have to identify the specific markers of the parasite, no? In a case of parasite, well, let's call it for cancer. Many cases, the origin of some cancers is a virus, no? And a question the virus can be a protein. Okay, so if you identify the protein, the presence of this protein, and some of these proteins we know have become inserted in our genome. So, and in some cases, for example, in papilloma and other, no, they become the cause of a given tumor origin. So, what we can do now is for sure with single cell data, analyzing the tissues of mm, people that are from biopsias that maybe not still developed tumor, but candidate tumor, you can see if the parasite cell or the parasite marker is there. Because at the end, you are reading three things at the same time, no? The samples, the genes, and the cells. So if you can match your question or protein of question or your cell of question with one of these, then you, of course, you will be able to do, or for example, to see if this specific marker is more intense in a given part of the human body. I think it's a very powerful tool, and I think
Yes.
yes, of course. The question is a lot of data, and you have to be careful to not create artifacts. and you have to understand what is the level of granularity of your, and for that reason we did all this work, not to try to understand how we can handle the tools and make use of them.
Okay. Thank you.
I don't know if there are questions from the attendees.
No, I don't think we have more questions. So if someone else want to ask something now, or if we don't have more questions, I think that we
Well, thank you everybody for attending. And well, at least you'll have my contact if you want to any question. We are keep doing a lot of work on this area, and in also to improve the methods of analysis of single cell data of different platforms as well. And
Okay, thank you
also-
very much. Yes, we take your data for sure.
Okay.
Thank you, Javier. Thank you very much.
Okay. So thank you. We can again stop sharing. And if nobody wants to ask in the chat, there are
Ya está cerrada la,